All right, everyone. Okay, thanks everyone for coming along. It's really nice to see so many people here. For those of you that don't know me, I'm Jamie Weir. I was doing recently a PhD at Ashworth here, and I'm kind of responsible for instigating these meetings. So if there's any problems, you know who to blame. Yeah, what else can I say? Just some basic admin things to begin with. So afterwards, we're going to be having refreshments. Those are up on the left outside towards the elephants. If you need to go to the toilet, it's down that way. And I'll hand you over now to Mark Watson from the Botanic Gardens here, and he's going to introduce Mark, the other Mark.
Right. Thank you very much, Jamie, and a warm welcome to you all here at the University of Edinburgh Medical Lecture Theatre. What an amazing lecture theatre this is. This tonight is a special event. It's a joint lecture between the University of Edinburgh and the Linnean Society, which I have the honour to be the president of. I must start by giving a big shout out to Jamie. He's very humble. You've been the driver behind these joint lectures for several years now, and you've really made them happen. 3. So we're looking forward to number 4, 5, 6. Yeah. Thank you so much for all the effort you've put into the organising, arranging an amazing speaker, and looking after us this evening. Thank you very much, Jamie. As I said, can I just ask, are there any fellows from the Linnean Society here this evening? Yes, we've got a few. Fantastic. Welcome to the fellows, and welcome to all of you. I hope that by the end of this evening, you'll be inspired to join the Linnean Society in its quest to be in a world where nature is understood, valued, and protected. The Linnean Society is the oldest society dedicated to natural history, the study of natural history. And we were formed in 1788, over 300 years ago, around the collections of Carl Linnaeus, and you can see those in the slide here that Jamie's put together. Over 300 years later, we're still pursuing the same mission, which, in those words, was the study of natural history in all its branches. Never before has our mission been so critical as we face the dual crises of biodiversity loss and the climate emergency. Our membership is truly international. Our members come from all over the world and range of disciplines, forming a community which is united by a passion for natural history. Although we're a science-based learned society, we're in fact a very broad church, and scientists, people who profess themselves as scientists, represent about half of our membership, and the rest come from historians, artists, conservationists, and people who are just generally enthused about natural history. If you're passionate about natural history and want to help the society in its mission, then please become a member. We'd love to have you in our society. Partnerships are critical in delivering our mission, and this is a great example of those partnerships, Jamie, here with the University of Edinburgh, extending the message out to a much wider audience. So thank you again for organising this. I'd also like to highlight our journals. We have 4 journals, and particularly amongst the younger members of the audience here, students, PhD students, please consider contributing your papers to those journals. The bonus is you might get a prize. There are 2 prizes for PhD work, and Jamie got the Marsden medal a few years back now, when you had to submit your thesis and have it judged. Nowadays, you don't have to do this. You contribute a paper to one of our journals, and journal editors pick the best ones, and you get either the Marsden Prize or the Irene Manton Prize. So have a look at our journals. They're great places to publish and get your work out to a wider audience. There'll be, as Jamie mentioned, there'll be a drinks reception after this lecture. So if you're interested in learning more about the society, then please come and talk to me or some of the other fellows who are in the audience. We'd love to tell you more about our society. But let's turn now to our speaker, Professor Mark Laxter. Mark is the head of the Tree of Life program at the Wellcome Sanger Institute, which is generating and analyzing genome sequences from thousands of species across the Tree of Life. But although this project has a global focus, you're really focusing down on the British and Irish species at the moment under the Darwin Tree of Life, and I'm sure you're going to be talking about that. And I'm sure many people here in the audience will know of that groundbreaking project. Many of you will know some of the background to Mark. In fact, there are many old friends here, in fact, and previous lecturers. Mark is well known to us here in Edinburgh. He's got a background of genomics of parasitic nematodes and has worked on a wide range of species from viruses to mammals, covering topics from the dynamics of infection of marine algae to the evolution of male plumage diversity in birds. Truly astounding range of things, Mark. You were at the University of Edinburgh for 24 years before you moved down to the Sanger. And since 2004, you were here as the chair of evolutionary genomics at the university. Personal chair, I think it's like that. And in 2014, you were elected to the Royal Society of Edinburgh Fellow. So it's my pleasure to be welcoming you back here to Edinburgh. I don't know if you ever lectured in this theatre before, so a new experience for you. And thank you for agreeing for speaking. So ladies and gentlemen, I'd just like to invite you to give a warm welcome to Mark, who will be giving us a talk entitled Busy About the Tree of Life, Genome Sequencing of Biodiversity. Over to you, Mark.
I'm used to shouting in lecture theatre, so I haven't shouted in this one. So thank you all so much for coming. Thank you especially to old friends, I guess I'd call you, colleagues, friends, who I haven't seen for a long time, and especially to others. So others who've come along to see what it is we might be doing. Can I ask a question? How to put? I'll make it. People reveal, please, if you know something. If you feel you know something about genomics, just to know. So those of you who didn't put your hands up, I'm going to be speaking to you, I hope. And so if I introduce a topic, I'm going to be looking at you and seeing if you're lost, asleep, or whether you can sort of nod that. Yeah, you get what I'm on about. Okay, I presume it's this one. Yeah. So we are, Tree of Life is a program at the Wellcome Sanger Institute, which is one of the world's premier genomics institutes. And one of the things we're really strong about is open science. And open science means releasing data openly so that all can use it, and publishing openly so all can read it. And in this case, also publishing my talk openly. So if you want to go through the slides on this talk again, it's all openly available at that tiny URL, toll-lin-soc 2025, or if you point your phone at the QR code, something might happen. I don't guarantee it'll take you to the right place. No, it will. It'll take you to the right place. Okay. So this is a view of deep space from the Hubble telescope. And what they did was they pointed their camera at the space between the stars and left the shutter on the camera open for a long time. And this is what they saw. So in the darkness between the stars were galaxies and galaxies and galaxies, as well as many, many stars. So each galaxy is hundreds of thousands of stars. And you can imagine that you could point this camera now, not the Hubble, the next one, at the space between these stars and look out to the edge of the universe. So there's an unimaginable number of planets and universes out there. And before the Hubble pointed its telescope between the stars, we didn't know what was there. So we're interested at the Sanger Institute in the Tree of Life program in sequencing everything. And it's not because we're museum collectors, although there is a little bit of that, I must admit. It's because we think by looking at the spaces between the things that have been sequenced already, we have got a genome sequence from the human. We have a genome sequence of a cow. We have a genome sequence of wheat. Looking at spaces in between, at the rest of life's diversity, we'll discover amazing things, just like the Hubble telescope discovered in the spaces in between. Okay, this is the only place we know where there is life. This is the famous blue marble photograph from 1972. This is the only place we know where there is life. This is the only place where we know that anything like intelligent life, if we are intelligent, given what we're doing to the planet, it's not completely sure, has evolved. And so this is an incredibly rich resource that we live on, an incredibly diverse and amazing planet. But as you all know, and as Mr. Darwin said, there's grandeur in this view of life. And this lovely quote that evolution has created the diversity on the planet. And this simple rule he invented, called natural selection, from so simple a beginning, life started somewhere, endless forms, most beautiful, most wonderful, have been and are being evolved. That's what we share this planet with, this amazing organic evolution. And he famously, also in one of his notebooks, made a little tree. He was actually thinking about the relationship between individuals in a population, but we can take this as a first estimate of what turned out to be the tree of life. This happens to be an oak tree in the grounds of the Sanger Institute, but amazingly, it's almost like Darwin's tree. Anyway, so our idea is to decorate the tree of life with as many genome sequences as possible. So we could have done that at any time, but why now? So we're linked to all the rest of life by this common thread being DNA. DNA is a relatively boring molecule. It's a very big molecule, but it just goes on and on and on. We represent the substructure, the subunits of this DNA molecule by letters, which indicate particular chemical side chains, basically T, C, G, and A. And this is what your genome looks like. This is what the genome of a fly looks like. This is what the genome of the oak tree looks like. They all just look like lots of strings of letters. But the important thing about these strings of letters is that they're not random, that they actually have in them the code, the instructions for making the organism from which they came. So some of the letters in this genome have no observable effect on what their organism looks like, but many of them do. And so from this raw piece of DNA, we can build a tree of life. So realizing Darwin's vision of building a tree of life. So you'll see this picture again several times. This is actually a picture of the organisms we've sequenced, and they range from the star-shaped thing up in the, top left, which is single-cell protozoan, right through other algae and plants, fungi, and all sorts of animals. Some of you who have memories going back to the 1970s, which is me, and maybe a couple of others here, may remember that we are linked by a common thread quote, because that comes from the United Nations conference on human rights. And there's a quote from that conference that came out. Nobody knows quite who said it, but we are linked by a common thread of humanity. And if we break it, we are all undone. So this is a call to look after each other. And it's the same with the idea of DNA as a common thread between us all. If we break this common thread, if we lose species through extinction, we're probably all undone. This is terrifying. And given what's just happened at COP30, it's even more terrifying. This is temperature change the last 50 years across the planet. There's a couple of small places on the planet where it's got ever so slightly colder, but mostly we are boiling our planet. And this is all largely anthropogenically induced climate emergency. And what we know is that all the biodiversity on this planet isn't just important in and of itself, but it's important because it supports us. So we may be consuming the planet. Here the Earth has had a great bite taken out of it by humanity, but we're actually supported by all this biodiversity. They provide ecosystem services to us all, which mean that we can survive. So trees produce oxygen and also sequester carbon. There's a very simple thing to do with the carbon cycle, but many other pieces of this biodiversity support us in ways which we don't yet fully understand. And if we break it, if we break this chain of life, if we break biodiversity, we're all undone. The planet is very diverse. So far we've managed to describe in the Linnean system about 1.65 million different species of eukaryotes. So these are species with nuclei. It's an absolute guess, and depending on whether you're an enthusiast for biodiversity or a relative pessimist. There's somewhere between 1 and 100 million additional species out there to be named. I've chosen a nice middle ground of that, which is 10 million. And new species have been described at 2,000 per year. So if you do the quick arithmetic there, it's been 300 years since Linnaeus. At 2,000 a year, and that's where we got after 300 years, 1.65 million. Even at 2,000 per year, it's going to take us forever to get to the end of the list of species to be described. So it's amazing. Most things are arthropods. I'm sorry if you're scared of arthropods or think they're nasty, no-see-em flying things, but they are actually the most important and diverse group on the planet. Nearly half of all species live in the soil, including in that the plants, which have roots in the soil. So soil is an incredibly important biome. And for those of us who've worked on parasites for all our lives, parasites are about 1/3 of all species. So if you include in parasites things that feed on blood, like mosquitoes and other blood-feeding organisms, but about 1/3 of all species that have been described are parasites living on other species. So amazing diversity out there, but about 150 are going extinct every day. So this is the biodiversity crisis, the extinction crisis. And this is just pulling some stuff off. You can tell I read The Guardian, pulling some stuff off The Guardian over the past couple of weeks. And it is truly terrifying. So we must act now. And I hope you all know that. And I'm sorry if there are any climate change skeptics or people who think it's not our fault, but I really do think we must act now. So I'm happy to have a long discussion about why we must act now. And I'm a genome biologist, right? And it's that old thing about if you give a carpenter a hammer, everything looks like a nail. Yeah. So to a carpenter, everything looks like, to a genome biologist, everything looks like it's got a genome. And I really believe that sequencing the genome of all living things on the planet will be a very positive act we can do to think about conserving biodiversity. So I think sequencing all these species on this tree is a very positive and conscious act towards conserving biodiversity and dealing with the extinction crisis. It's not my idea, not my idea alone. It's an idea shared by people around the world. So this is the top page of the website of the Earth Biogenome Project, which is a project started 7 years ago now, which proposes sequencing life for the future of life, proposing sequencing all of the species on this planet. The Earth Biogenome Project is a network of networks, a collaboration between projects all over the world, some of them focused on individual groups of organisms, just plants or just butterflies, some focused on particular areas of the world. We've got a particular project I was mentioned on Britain and Ireland, or on particular ecosystems or biomes or ways of life. So we have an aquatic symbiosis project. Okay, so the Earth Biogenome Project is the umbrella project, which is encouraging people all around the world to sequence. And the Earth Biogenome Project now has about 65 member affiliate projects around the world doing genomic sequencing. At the Sanger Institute, we were really pleased to be able to join the EBP and are now a major contributor to EBP goals. These are two quotes, one from Harris Lewin, who this is from a paper we published just a few months ago, where he said, yes, the biodiversity crisis is gathering pace, but that just means we've got to sequence faster. And there's a quote from some Mark Blackster about preserving blueprints for future generations. So what do we think these genomes will do? So first of all, for conservation per se itself, we think these genomes are going to give us a new set of tools with which we can monitor where an organism is, who it's interacting with, and then also use that data for better conservation. One of the things that's really clear is that, for example, introducing animals back to the wild from zoos can be very difficult because the genetic diversity of the animals collected in zoos, little bias there from Mark about what he thinks about zoos, oops. Anyway, the animals in zoos is a subset of the genetic diversity out there. So you've got to be really careful about how you breed those animals in zoos and what you put back. So are we restoring genetic diversity in the wild? We can use DNA to monitor diversity. In this room at the moment, sorry, you're breathing in skin cells from everybody in the room at the moment. That's a bit, isn't it? Sorry. If we took a big vacuum cleaner and sucked all the air in this room onto a filter and extracted the DNA from that filter, we would be able to find out what was in this room. At the moment, I think it would be mostly humans, but we'd also find fungi and bacteria and viruses in the air. So you can use environmental sampling to tell who was there. If you do this on water in a river coming out of a forest, you can tell what's in the forest based on the DNA that's from the environment in that sample. So we can use the complete reference genomes to be able to say this species was here then. We can also say something about the genetic diversity of that species. We think that these new genomes are going to help us move to a post-oil economy. Unfortunately, COP30 didn't quite manage that, but we are going to have to move to a post-oil economy. And so where are we going to get our plastics from if we don't have oil to make plastics? Where are we going to get our nylon from to make nylon ropes and threads? And nature's done that already. Spiders spin silk, and different spider silks have different tensions, have different strengths, up to the strength of Kevlar. They have different elasticities, they have different stickiness. That's all based on the sequence of the genome, which makes proteins with different sequence, and the proteins go and make up this silk. So if we have a whole library of spider silks, maybe we can engineer bacteria or yeast to make nylon for us, or spider silk. That's already happening. But imagine if we had all the spiders and all their silks, we could pick from a library of silks and decide exactly what it was we want to make. All our antibiotics that we use today are ultimately derived from biological source, either fungi or bacteria. And so by sequencing many, many more fungi, we're going to discover new drugs, the enzymes that make new chemicals, which we can use to treat new or old diseases. So there's lots of diverse new biologicals and biomaterials will come out of this project and be able to be accessed by a new bioindustry, which is not threatening the planet as our petro industry does now. And then for those of us who are scientists, we're going to get a new understanding of life. So to date, well, before we started this project, there were very few genome sequences available. There was ours, there was mice, there was cows and sheep. A very small subset of species that had their genome sequenced. And so what we knew about how organisms made their life, how they organized themselves internally, if you like, to deliver the phenotypes and the biology we see, was actually hidden to us. And as we're sequencing more genomes, we're finding more and more amazing things about how organisms organize themselves to produce the diversity we see. So what we want to do is build an open lending library of life. We want to make a library that's got Linnean catalogues. That's what the Linnean Society and all the people who've been working under Linnaeus ever since have done, is given us a catalogue to what should be in the library. And we want to make a library of life where you can go to the librarian and say, can I have the book of the badger? And the librarian will say, yeah, here it is. And you say how much that will cost, and they'll say nothing. And how long can I have it for? And the answer is forever, because we've just made a copy of it for you. And you can go off, and if you're interested in badgers, you can use that badger genome to understand the social structure of badgers, their susceptibility to TB, their relationship to other badgers and other carnivores, etc. But we want to make a library of life, an open lending library of life, which has an entry for every single species. And if you're interested in it, you can take it back. There's also no limit. I used to be really frustrated when I was this high, going to the library and you could only take 6 books out, which meant if the series had 8 books in it, I could take the first 6 out, and then by the time I came back, the next 2. But you can take out as many as you want. So an open lending library of life, which then we think will boost understanding, bioeconomy, including medical benefits and conservation. So we have a Tree of Life program at the Sanger Institute. We are 100 people now. Started off with me. And we've got 100 people now, amazing people who are incredibly dedicated to what they do. It's everything from administrators, lab people, clever bioinformaticians, and people really good at organizing stuff. So database people. And we have a whole bunch of major projects. The flagship one is the Darwin Tree of Life project, which we aim to sequence all the species that live with us on these islands, this archipelago off the west coast of Europe, which has the human nations of Britain and Ireland in it. The reason for choosing that is because the biota of our islands is so well known. We've had wonderful natural historians going all around the islands forever, it seems, identifying who is where. And so we have a really good catalogue of what is there. And it says here, phase 1, 8,000 species by 2026. We won't hit that. There was this COVID thing happened, which happened 6 months after we started the project. So we had a slight delay, but we will reach 8,000 species by 2027, I'm pretty sure. So that's everything that lives here, whether it's a mammal, a bird, a fish, a frog, all the plants, the mosses, the lichens, the fungi, and all the protists, all the single-cell organisms. We've involved a bunch of other projects. I'll just mention aquatic symbiosis genomics. So this is a project which is global rather than just Britain. And it's looking at organisms that live in symbiosis with each other. So these are organisms which separately can't live, but when they're brought together, they make a new organism, like lichen or like coral. These are organisms which collaborate with each other. So we're looking at those in marine and freshwater systems. And for those of you into Lepidoptera, which Jamie is, we have Project Psyche, which is sequencing all the Lepidoptera of Europe. So that's 11,500 species. We just hit 1,200 of those. So all 11,000 species. And that's to look at them for their intrinsic worth, but also because many of them are pests and pathogens of various crops. Also because they're canaries in the coal mine for climate change in Europe. They're used for monitoring of climate change, etc. So we're involved in lots of projects which are generating lots of genomes. I'm going to focus a little bit on Darwin project. And we stole again another 1970s slogan. It should be act locally, think globally. So we said sequence locally, think globally. So on these islands that we share all these with all these animals and plants, there are about 70,000 species, but that corresponds to about 40% of the family level taxonomic diversity of all species in the world. So it's 70,000 species out of 1.7 million. But species are grouped into genera, and genera are grouped into families. And if you look at the family level, we've got 40% of all the families in the world living in and on and around these islands. So if we can manage to sequence the species on our islands and in the seas around, we can probably sequence anything. So we use this as a demonstration project to say, if we can do this, anybody can do this. The other reason we decided to sequence locally is for, if you like, ethical or political reasons. It would have been great to imagine a Tree of Life project which had a summer station somewhere in the Caribbean where we could go and collect wonderful reef fish and generally do really wonderful biodiversity stuff. And maybe in the autumn, we'd head down to the Philippines. And that's absolutely classic, what's called helicopter science or neo-colonialist science. We wanted to be very clear that we were sequencing species for which we had, if you like, the ethical and moral right to sequence, and where we could demonstrate really good citizenship in the global world of sequencing things by making these sequences then available to the whole world. So obviously, if we sequence a golden eagle, which we have, golden eagles are found from Kamchatka all the way around and back to Kamchatka via Scotland. The golden eagle we sequenced happened to be a piece of roadkill. It was hit by a truck. But that sequence, that golden eagle sequence, is now available to anybody studying the biology of golden eagles around the world. So we're using our local fauna and flora and fungi and protists to illustrate how we can generate this data for the whole world. When I say we, it's not just the Sanger Institute. We're a collaborative group, and we involve biodiversity partners. So these are people who work in the museums and botanic gardens, and Zoological Society of London for the zoo, and also, importantly, places like Wytham Woods, which is a field station near Oxford, run by Oxford University, which has been a long-term ecological research site, and now we're sampling from it, so it's becoming a genome research site. And we have partners who sequence, that's ourselves, and the Earlham Institute, and then we have partners who do analysis. That includes the European Molecular Biology Laboratory, European Bioinformatics Institute, EBI, but also others on this list. So we're a big project, all funded by the Wellcome Trust. So this is one of the first projects Wellcome Trust, which is known as a medical charity, has ever funded in this area. And that's because the Wellcome Trust recognizes that biodiversity supports human society, and therefore biodiversity loss is a risk to human health, human societal health. So what we built was what we call the Genome Engine. And the Genome Engine, I'm going to take you through some of this. This is where I'm going to teach you all to become genomicists. And you're all going to understand this wonderful green snake diagram. Okay, first thing is we find the snail. Entertainingly, Cornuaspersum, it's a common garden snail, gets a lovely Latin name. We also do what's called DNA barcoding. So we sequence a little bit of its genome, which acts as an identifier sequence. That, first of all, assures us that we caught the right thing. Garden snails, it's quite easy to tell them apart. Small moths, not so. But also allows us to track it through our systems. And we find out where we found it and who found it, and anyway, all that sort of stuff. Then we extract the DNA and RNA. So the RNA is the first product from the DNA when genes are expressed. And then we do the sequencing. So we do three kinds of sequencing. One is the transcriptome, called RNA-seq. This is where we're just sequencing the expressed genes, so we know which bits of the genome turn into messenger RNAs and then get turned into protein. But then we do two different types of sequencing. One is called long-read sequencing, and the other one is called high-C, and I'm going to take you through those in a sec. We then do a primary assembly, a scaffolding, and a curation, and that turns all this mess of data into beautiful chromosomes. And that's one of the things we're doing. We're trying to only release chromosomally complete genomes. It would be easy to throw the snail into the sequencer and get out a pile of data and just dump that data, saying, here you are, make of it what you will. We want to prove that the data we give are good. So that's why we only release chromosomal genomes. We then submit it to the public databases, at which point it's open to everyone. We find where all the genes are, and we publish each genome as a little genome note, which says that Jamie found this, Mark identified it, and this team did the sequencing. Here you are, there you go. So it's a way of everybody who worked on it being credited. So I'll just go back to beginning here, the long read bit. This is the last page of The Origin of Species cut up into small pieces. For those of you who are librarians or worried, I did not cut up a real book. This is digitally cut up. So this is what we get out of our long read sequencer. We take the genome and we've cut into pieces which are long. They're about 20,000 letters long, but it's still not a whole chromosome. A whole chromosome can be 100 million letters long. And we get a pile of shredded genome. And what we can do is we can use the text, the A's, G's, C's, and T's, to line all this up and recover, as if by magic, page 490 of the conclusion, there is grandeur in this tree, in this view of life. So from long reads to an assembled piece of the genome. So because we know this is a book, we can look in the top corner there and say, this is page 490. This comes after page 489. Yeah. And so we know where to put this. If this was a book, we'd know where to put it. But in a genome, we don't know where to put this piece. Yeah. We don't know where it comes in the list, because we don't have the same sort of index typed onto each piece of genome. And this is where the magic of a technology called chromatin conformation capture comes. And this, I'm going to take you through this, and this is where I'm going to be staring at people who aren't genomicists to see if I can persuade you that it's interesting. So here's the human chromosome set. So 23 chromosomes. This is a female, so it's got a double X. And there's a way of painting chromosomes under the microscope such that each one shines with a different color. It's very clever. Can go into details how it's done. But here, you can see what's called a metaphase spread. So when cells are about to divide, all the chromosomes condense, and so they all separate from each other, and then you can stick them on a glass slide and stain them with these beautiful colors. And you can see that you've got pairs of chromosomes. Here's the two X's here. So, hooray, we can paint chromosomes different colors. What if you do that in a nucleus that isn't just about to divide? So these are nuclei in just normal cells which are doing their normal business. They're not about to divide. And what's really exciting is that those chromosomes occupy territories inside the nucleus. So this is the nucleus of a human cell, stained with the same set of paints used to paint the spread at the side. And I hope you can see that, for example, that red set, which presumably is the X, is in one place in the nucleus. So in the three-dimensional space, if you imagine this in 3 dimensions, the chromosome occupies a little territory inside the nucleus. And it doesn't matter what nucleus, if you look at 100,000 nuclei, the X chromosome will always be in a little domain inside the nucleus, close, all wrapped up in itself, and relatively separate from all the other chromosomes. So these are called domains. And so there's a model of what that looks like, and you can label every chromosome using that. So this discovery that chromosomes lived in domains immediately gives us a way of joining the pages of the book back together into chapters. And so what we do is we fix the nucleus using a chemical that freezes that structure. We cut the DNA with an enzyme that just makes a cut. And then we join the DNA together again. So in most cases, the same bits of DNA join together again. But sometimes it's a bit of DNA next door, right? It's called proximity ligation. So ligation is joining and proximity just close. So the red chromosome will be more likely to join to other bits of red chromosome. But it might be a piece of red chromosome from one end and a piece of red chromosome from the other end that get joined together because it's all wound up in this domain. So it's called chromosome or chromatin conformation. So it's the way they confine sequencing. It has lots of names, and it ended up in the literature as being called high C, which is just high throughput chromatin conformation capture. And what this means is we can take our little pages that we've assembled that pretend those red squares there along the diagonal are each of those pages out of the book that we say we're pretty sure this all goes together because we were able to line up all the text. And we use this chromatin conformation capture data to sort out all this mess of things off the diagonal until everything is joined up. And so this chromosome conformation capture or high C sequencing allows us to turn the fragmented stuff we get out of the sequencers into chromosomes. Okay, this is called the high C plot. And you're going to become experts in high C plots in the next 4 minutes. Okay, there you go. So here's showing it working across animals and plants. Here's a dolphin. This genome is 3 billion letters long, and you can see all the chromosomes are joined together. Up there is a butterfly. Its genome is about 500 million letters long. All of its bits of DNA joined up beautifully. The one at the end that has that extra signal at top and bottom on the butterfly, that's the sex chromosome. Happens to be a Z chromosome in butterflies, which has lots of repeats on it, which are also found on other chromosomes, so you get mistakes in the mapping. You see lovely patterns in the apple, for example. There's lots of signal off the center. That signal off the center is actually very informative. So here's the biggest genome that has ever been sequenced by and assembled by anyone. It's from mistletoe. So the ordinary mistletoe that you'd have hanging at Christmas, New Year. So it's a parasitic plant. It has a genome which is 30 times as big as the human genome. Don't ask me why. It needs 30 times as much DNA as the human genome. It only has 10 chromosomes. and I'm sorry about the changing color scheme. The color schemes always change on these things. This was in red and white. This is red to blue. You can see there's 10 chromosomes along the axis. Each chromosome is a cross. These chromosomes are so big that the way they're organized in the nucleus is that each end is attached to the outside of the nucleus, and also a bit in the middle called the centromere is attached to the outside of the nucleus, so they kind of loop into the middle of the nucleus and out again. And so the two arms of the chromosome are next door to each other. And that means every single chromosome turns out as a cross. And because all the chromosome ends and the chromosome middles are attached to the outside of the nucleus, they also tend to cross over each other. That's why you get these crosses off the diagonal as well. So that tells us where the centromeres, the bits of the chromosomes that organize their division, are located. And so this is a lovely tartan pattern. It's available from a shop near you on scarves and blankets. Okay, and then we get exciting things. So here's the rose. So Gertrude Stein, 1922, a rose is a rose is a rose. She actually got it wrong. She should have had another 2 on that. So it's a rose is a rose is a rose is a rose is a rose. You and I have 2 copies of each chromosome. Roses have 5 copies of each chromosome. They're called pentaploids. And so in this case, you can see there's a bunches of 5 chromosomes with signals off the diagonal. I've zoomed in on the next slide. This is one chromosome, one type of chromosome in rows. And there are 5 copies of it. And there's similarity between the 5 copies. So you get one official copy, one that's very close to it, and then 2 others. 3 others, rather. So 5 copies of each chromosome. So we're assembling not just each chromosome, but we're assembling every copy of each chromosome. It gets even more wonderful. It gets crazy. Here's hornbeam, which is octoploid. So it has 8 copies of everything. And there you are, 8 copies of everything. So you can understand this high C plot now. The strong signal along the diagonal is the chromosomes folding in amongst itself. The signal off the diagonal is because those chromosomes occupy similar territories. So high C plots like this are very pretty. As I say, you can get them as wraps and throws, but they really help us turn our raw data, that chopped up page of Darwin. We can assemble a page, and now we can put all the pages in the book in the right order and say, here's chapter 1, and here's chapter 2, here's chapter 3, here's chapter 4. And so with that, we can do genomes at scale. So all well and good, brilliant, it all works perfectly, except we're taking organisms from the wild. And organisms in the wild come with lots of free other organisms inside them. If you think about it, on you are fungi. In your gut are bacteria, on your skin are bacteria. If you happen to be a wild organism, you might also have a parasite growing in you somewhere. And so when we sequence these things, we get a soup, a monster soup of things. This is supposedly Thames water. I hope Thames water isn't that like this was some time ago. Thames water. But it's much the same with what we find when we pick something from the wild. We get what we call its cobiomes. We don't just get the organism, we get a little snippet of the ecosystem it came from. And so we get the black box is what we get, and what we try to do is do a shape sorter and sort out all the different genomes so that we can say we've sequenced a moth in this case. We've got its beautiful high C plot. We've got its mitochondrial genome, which is the genome that lives inside the mitochondrion, which is inside of almost all eukaryotes. And in this moth, there were 3 different alpha proteobacteria, bacterial parasites, which are transmitted, in this case, transmitted from mother to their offspring. So we've got 5 genomes instead of just getting 2. And so sometimes things are really easy. This is a pincushion moss. This is called a blob plot for no other reason than it's a blob plot. Sujay Kumar, who's in the front row here, is responsible for these in some way. The blue dot in the middle is actually a whole lot of blue dots all piled on top of each other, which are all the chromosomes of this moss. And they all have the same proportion of G's and C's in their DNA. So GCAT, they all have the same proportion of G's and C's, and they all have about the same coverage in our sequencing data set, which means that when we sequence them, they're all about the same proportion. So it means they're all from that organism. There are a few other dots here, which are possibly the bacteria that will live in this pincushion moss. Moss are great to sequence because that's a haploid organism. There's only one copy of its genome in each cell. Here's what happens if you try and sequence a sponge. No, this is a ciliate. Sorry, I'm lying. This is a ciliate. I took the sponge out. This is a ciliate protozoa. So this is just a tiny little single-celled animal, but it likes living with bacteria. So all that horrible schmear there is actually all bacteria. The ciliate is this little bit here, this little blue bit off the side. So in this case, we've got 100 and something bacterial genomes as well as the ciliate genome. So we're sequencing ecosystems. Here's the sponge. So 10 nuclear chromosomes, 1 mitochondrion, and 127 bacteria from all around the tree, including archaea. If you know anything about bacteria, that's a really separate group of prokaryote organisms. The other thing we tried to do is sequence small things. So most of life is small. We're weird because we're huge, and we're really interested in huge things. But actually, most of life is tiny, and most of life lives in between sand grains and soil grains. This is from a beach, and there's nematodes and copepods and other things in here. And we developed a method that can take one nematode, which has got about 1,000 cells, and turn it into a genome. So we're really proud of this because this suddenly means that we can sequence everything. We really can now sequence everything. So there's an elephant, the smallest known, not to scale, the smallest known vertebrate, tetrapod rather, which is a tiny frog. And this is the nematode we sequenced, which is 1 millimeter long, but not very thick. It has about 1,000 cells. And lo and behold, you've seen that before. That's a lovely high C plot. Those nice chromosomes. Awesome. So we can sequence anything. And so this afternoon, we got to 3,500. So we expect in January, we'll get to 4,000 genomes. And this is everything from, as I said, from single-cell protozoa, algae, mosses, plants, the sort of higher plants, fungi of all sorts of flavors, jellyfish, mammals, mollusks, annelids, nematodes, of course, and then a whole bunch of arthropods. So it's quite astonishing. So our 4,000th genome will mean we've got to about 100,000 chromosomes. So we've sequenced 100,000 chromosomes. And it's about 1,000 times the length of the human genome. And it's taken about the same amount of time, and so far has cost about 1/10 of what the human genome cost. No, less than 1/10, 1/30 of what the human genome cost. So it's quite amazing. So which time is there? I'm just going to quickly skip through my favorite, at the moment, organism, which isn't a nematode, but it is a butterfly. So there you go. So is that all right, Jamie? I can have 5 minutes. So why 23? Why not 22, 24? Why not 100? Why? What controls the number of chromosomes in an organism? The way we found that out is not by looking at other humans, because we're all probably going to have about the same, but by finding organisms which are really extreme in terms of how many chromosomes they have. So Lepidoptera, butterflies and moths, are relatively boring. Most of them have 30 or 31 chromosomes. And if we look at them, because we have so many genomes now, we look at them, it's the same 30 or 31. So they're really deeply conserved. This is 270 million years of having the same number of chromosomes and the same genes on the same chromosomes. There are a whole bunch of species which have slightly fewer. That's usually because they've just fused 2 together or 4 together. But I hope you can see that this graph goes way out to the end. And there's a number here at the end saying 223. So that's this. This is the Atlas blue butterfly from the mountains of Morocco, not a detoll species, I should point out, which this is the karyotype of the male. And there's lots of little dots, which have got red dots on top of them. They're actually just little grayish-black dots, and 2 big splodges in the middle. So that was counted as having 223 chromosomes. So of course we had to sequence it. So Martin Gascoigne-Piz, who's a renowned lepidopterist, had collected a single female specimen in a completely legal collecting trip in Morocco 15 years ago and had stored it in a freezer. And from he'd given it to his colleague Roger Villa in Barcelona. Roger and he transferred it to a freezer in Oxford, and Charlotte Wright, whose project this is, drove over to Oxford and picked it up from the freezer there and brought it back to us. This is one poor little butterfly which had a 15-year journey getting to us. And then we sequenced it. So you've all seen high C plots. Is this a good high C plot? Nothing seems to be joined together. Possibly the ones up at the top, possibly, but the rest is just dots, right? This is actually true. So Polyomytus atlantica doesn't have 223 chromosomes, it has 229. And every single chromosome is tiny. So this is a picture of all those tiny chromosomes. It has 227 autosomes and then 2 sets of sex chromosomes. So it has 2 different Ws and 1 Z. Sorry, 2 different Zs and 1 W. So butterflies are not like us. It's the females that are heterogametic. So we're XY, butterflies are ZW. And so during the evolution of this species, every single chromosome that its ancestor has been cut into 10 pieces. Every single chromosome, except for the sex chromosomes, which are still exactly the same size as they were in the last common ancestor. It's got 2 Z chromosomes. It would be a bit like us having 2 X chromosomes, 2 different X chromosomes. That's a particularly a weird thing that butterflies sometimes do. They generate Z chromosomes from fusion of ancient autosomes, but they're all big. Okay, so this individual is now the single organism with the highest number of chromosomes known in a diploid organism, this poor little butterfly. We don't know whether it's a weird one. Maybe normally they have 223 and we just have to catch one that has more. Every single one of those chromosomes is 2 million letters long, which is tiny. It's tiny. The whole of the Hox cluster is on 1 chromosome, Vernon, you'll be glad to know. So Vernon taught me about Hox clusters when I was an undergraduate. And these are genes which pattern the nose to tail organization of most animals. And still in this butterfly, it still has an intact Hox cluster. Okay, so if the chromosome number equals the frequency of breaks and the frequency of joins, that's what determines the chromosome number. Usually there's no breaks and no joins ever. But we now have a species which has many, many more breaks than joins. And we can go in and ask of that species, what went wrong? Oh, it's not gone wrong because it's still alive today. What do you do differently from your friends? What's changed in your molecular mechanism? And by seeing what's changed, we can then understand what it is that's conserving chromosome number in all these other species. So we actually have a whole set of butterflies which have weirdly high chromosome numbers, and we're now looking at them to see if we can find out the mechanism. Okay, so that's just one of the little bits of new biology. Crazy organisms give you interesting access to information. This is a high C plot for Christmas. We have no idea what this means, but it is true. This just came out. I've turned it on its side. Anyway, that came out 2 weeks ago. Somebody posted it on our Slack channel saying, it's Christmas. Anyway, so I get to talk to you, and I hope you can hear that I love the job I got. It's a dream of my life to do this, as people who know me know. But really, I can only do this because of the people we work with. So all the people who collaborate with us, especially those that collect samples, for us is really important because we can't collect everything. And then we have these amazing experts in sequencing at Sanger, both in scientific operations and the Tree of Life teams. And then my team, I put Pablo, Erna, and Lewis here because I did have some stuff about nematodes in, but I put Charlotte in at the last minute, and Charlotte, who's a PhD student with me. Okay, so thank you very much.
All right, thank you very much, Mark. That was really great. Do we have any questions now in the room?
Especially from people who aren't card-carrying genomicists. Do you understand what high C plot is now? Somebody know. Yes.
Mark, could you just repeat any
Yeah, I'll
questions
repeat
for
the question.
the online
Does the material have to be fresh frozen, or can it be pickled in alcohol? The best results are always from fresh frozen material. And we like stuff that's been snap frozen into liquid nitrogen. So really, and that captures that structure of the nucleus, if you imagine. We have done some sequencing from alcohol preserved specimens. And in, for example, invertebrates, which have nucleated blood cells, birds and amphibians and fish, often we get very good data from alcohol fixed blood. Arthropods, the story is slightly more fuzzy, but we discovered that if you gently squish the arthropod as you put it into the ethanol, which presumably just lets the ethanol get in more efficiently, the results are better. So I think we will be able soon to be working from ethanol fixed specimens of many, many taxa. Yes. So the question is, can we link major innovations in evolution with whole genome duplications? So this is true in some cases, but not others. So yes, it's very true that, for example, in the vertebrates, we are the result of 2 whole genome duplications. We have 4 copies of some genes, 3 of others because we've lost some. So yes, that is still true, and we can see that. These species that we see, which are polyploid now, for example, those plants I showed you with 5 and 8 copies, they are very recent duplications. So we wouldn't say that they were doing anything particularly novel. It's something to do with the way plants do meiosis, cell division. Interesting that the mistletoe, which is the biggest genome of all, it's not quite the biggest genome of all, there are 3 genomes bigger than it, known to be bigger than it, it's the biggest one sequenced, is not polyploid. It just has 10 chromosomes, and if you count the number of genes, it's just the number of genes you'd expect of a plant. Most of that 90 gigabases, so 30 human genomes, is repeat, 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 repeat. And so I don't think much innovation biologically is going on there, apart from the fact that it's a very nice place for repeats to live. There are some exciting things with chromosomes. I talked about chromosomes just while you got me. Earthworms, clitolate annelids, for those of you who are, and you get marine annelids, polychaete worms. One of the things that really separates the terrestrial worms from those marine ones is that the terrestrial worms have rearranged their genomes completely. So they haven't duplicated anything, but they rearranged it all. So the marine ones tend to have 18 chromosomes, and the clitolates have different numbers, but they're all chip-chops, cut and glued across the way. So if that had only happened once, we'd say, well, that just happens. But it turns out it's the same in cephalopod mollusks, so octopus and squid and things, compared to other mollusks. Other mollusks are boring, like most leps are boring. Sorry, Jamie. Most lepidoptera are boring.
This meeting is over.
But cephalopod mollusks have chopped and changed their genome, completely rearranged them. And both of those are important innovations. So cephalopods, well, octopus are very different from clamshells, and terrestrial earthworms have invaded a new habitat. So there is a strong signal of major genome rearrangement at major events in evolution.
There's still quite a few more to do.
There are. So the question is, how long are we funded for? And will we ever get to the end? Yeah. So the Wellcome Sanger Institute is funded by the Wellcome Trust, and we have an approximate 5-year horizon on funding. So at the moment, I'm pretty confident at 5 years, and I've got to work hard and persuade the Wellcome Trust that they should continue to fund this. We want to up the rate at which we're sequencing, because obviously, even at 2,000 a year, we're never going to get there. I mean, yeah. So what we're trying to do is up our rate of sequencing. So we want to be up at 5,000 a year in 4 or 5 years' time. We'll see if that happens. The main problem there will be collecting specimens, collecting species. It's hard to find things. Once you've got all the big things and the things that don't run away, then it's hard to find the others. The other one is we want to make sure that other nations, other jurisdictions start sequencing at the rate we do. So we have a vision of founding, let's say, 30 or 40 additional large-scale genome centers across the world, including in centers of high biodiversity, so including in the global south. Trying to do that again through philanthropic funding, giving sequencing centers to projects that want them and helping them run them at scale. So yeah, it's a huge job.
Big
Sure. How do we collect things? So, yeah. So we collaborate with biodiversity specialists. So the museums, the Botanic Gardens, the Naturalist Museum, the Botanic Gardens, people in White Woods, the Marine Biology Association, and they know where to go to find things. Right, so that's the first thing. There's a lovely story. One of my postdocs works on isopods. There's a particular weird isopod that lives on a beach in Cornwall, and it's like, okay, it lives on a beach in Cornwall. That's not going to happen. A beach is a big place to find a little thing that's about 3 millimeters long. So she went with the guy who described it. He's about 70 something, and they stood at the top of the beach, and he said, "I think it's under that rock," because he could remember from having been there as a PhD student. And the rocks, and sure enough, they found it, and they spent the rest of the day having ice creams and going for a walk. I mean, it was like there was going to be this hard day on the beach, digging the sand, trying to find this thing, and she found it in well, half an hour. So we rely on those experts to find them. They are identified on site as much as possible. We take macro photographs of the taxonomically relevant bits, and then we snap freeze them. When we snap freeze them, we pull a little piece off, for if it's an arthropod, it'll be a leg. We stick it in a separate tube, all properly labeled, and that's sent for DNA barcoding. And then you get back to the lab, you've got all these things, and then a competent taxonomist looks at the photograph and the description and decides whether or not this is the thing it says it is, and then it comes to us. And we still get a lot of taxonomic rearrangement and interesting things happening, and I've misidentified a few things myself, and so on and so forth, but there's a check on that. So we try and be really respectful of landowners' rights. So if there's a species on your land, it's your species, of natural history law and custom. So if we're sampling from anywhere that has any sort of designation as a site of special scientific interest, we have special permission. And if we're collecting a species which is known to be endangered, we have extra special permission for that species. So we try to be as ethical as possible up front. Yeah. It'd be lovely if people, I mean, we have had instances. A famous Irish bat arrived in the post one day without collection information. It's an endangered bat, so it should have had a convention on the trade of endangered species. Should have had a CITES document with it. It should have had a veterinary certificate with it saying it was rabies free. So we were left with this illegal bat, which we had to do a lot of big sorries to various people in various institutions to finally get permission to keep it. Otherwise, we would have had to incinerate it, which seems wrong. Anyway, so don't just send us frozen bats, please. Yeah, yeah. So she asked, do I really mean that we can sequence a single nematode? And yes, we can. We can sequence single larvae. The success rate is slightly lower, but for example, for larvae of parasitic nematodes, strong-gall nematodes that infect sheep and cows, and we're sequencing, where's Josephine? We're sequencing the Kilda poo nematodes. Yeah, so Josephine works on a wonderful study system on St. Kilda, where there's sheep, and the sheep have nematodes, and the way you get at the nematodes is they drop out the eggs in poo, and you take the poo and you culture the poo nice and warm for a while, and then nematodes develop from eggs to third stage larvae, which are about 0.5 millimeter long and have about 500 cells. And we can sequence a whole genome from one of those nematodes. So we have, you don't know this yet, we have 8 or 10 Kilda nematode genomes. So yeah, we can sequence single worms and single tardigrades and single decipherings and single chinorhynchs and any other weird, weird tiny critter you want to work on. Yes. Oh, will sequencing technology change in the future, and will we get to do it in the classroom? Yes, there is new technology coming up all the time. At the moment, there are 2 different long read technologies. You probably use the MinIron. Okay, there's 2 different long read technologies. One you use to get those strips of paper which they can assemble into the page. Those are going ahead leaps and bounds. So I don't expect there'll be anything super new in the next 3 years, but who knows after that. But for the technologies in humans in sequencing are really driven by what sequences humans really well. For some reason, we're really interested in sequencing humans. Discuss. But so there are lots of technologies just coming out which are really speeding up and dropping the cost of that, but they tend to involve very big machines, which probably won't get to the classroom soon. But yeah, the Chilean 1,000 Genomes project sequenced 2 earwigs in the classroom with students in Chile using MinION. So I think it's a lovely, lovely project that they did, because they got the students to choose what they want to sequence. They want to sequence earwigs, and the people around the project said, "Oh no, what are we going to do?" And they did. They sequenced them, assembled them, and published them. So yeah. So are these folks from your class? Excellent. Excellent. Well, there'll always be a job for you at the Sanger Institute. So it'll never, genome biologists never run out of jobs. Hi. So given that we're trying to get to 1.7 million, will we ever get there? But in a much deeper philosophical way. So honestly, we won't. So I'm not saying that my job is pointless. I'm just saying, honestly, we won't get to 1.7 million. So for example, in Britain and Ireland, we have 72,300 odd species have ever been identified on these islands. But if you ask how many have been seen since 1990, the answer is 30,000. And it's not that the other 40,000 are extinct. If you look at what's been described, they were incredibly rare in the first place. Okay, so they've been seen once or twice. And so the chance that we will chance upon them again is quite small. So I think actually the accessible sequenceable species group in Britain and Ireland at the moment is about 30,000 for the technologies we're using. So how do we move beyond that? So I said most things are small. And so we have to actually change out the way we look at things, the way we do things to respect that small size. And so, for example, one way of doing it would be sequence first and ask questions later. So to go to the soil, find all the microbiota, the myobiota, all the small animals in the soil, and generate some sequence data from every single one that you see. So that might be 5,000. And then afterwards go in and say, which one of you 5,000 are the same species? And do we know what you are? Right? And that might deal with, the first 300 might all look like the same species and we know what it is. And maybe we have a genome anyway, or if we don't, we sequence one of them. And so I think we might have to do it like that, and a sequence first, ask questions later program. I've mentioned DNA barcoding a few times. So DNA barcoding is where rather than sequence the whole genome, you sequence a little piece, and it acts like a barcode to identify that species. So there are all sorts of problems with DNA barcoding, and many taxonomists don't like it. But it is undoubtedly true that DNA barcoding is a very quick way of sorting organisms into bins. So the International Barcode of Life has a database called the Barcode of Life database. And they have something like 20 million specimens, DNA barcoded. And these now correspond to 1.2 million of what they call bins, which are just groups of sequences which are all similar to each other, but different from all others. And so they're species-like objects. And they think they're going to pass the Linnean taxonomic number sometime in the next 2 years. So they're adding bins at something 20,000 a month. So it's an astonishing project, but all it does is tells you this is the diversity of the gene that they use in life. So I think that's probably the way we'll go, that we will barcode things. So sequence first, ask questions later, and say, of this group of flies, tiny little black flies, of this group of flies, there are 5 clusters. So we're going to sequence 1 from each cluster, and we're going to call them species, whether or not they have Linnean names or not. So it is yeah. I guess the goal is unachievable, but it's worth, if we keep that as our stretch goal, then it's really worth trying to get the technologies together that we can approach it.
How do RNA viruses fit the tree of life?
How do RNA viruses fit into the tree of life? The answer is everywhere because they're inserting themselves all the time. But so we're not, so I should just say we're not trying to sequence bacteria, although we do by accident. I'll show you the sponge example. And we're not trying to sequence viruses either, although we do by accident. So we're having a talk at the Sanger Institute in 2 weeks' time from someone who's just been screening our insect data sets and has something like 7,000 new viruses. So these are the cobiome that just comes along for free. So we are sequencing these. How do they fit into the tree of life? Well, I think viruses probably have multiple independent origins from, yeah.
The summary of all the sequences in the tree of life, what does it tell us about the origin of life? Has it arisen once? Twice?
And given that all life works off DNA and uses approximately the same genetic code, so this is a way of translating the 3 letters into amino acids, I would say that it evolved once. Viruses are things which can evolve. Given life, a virus can evolve de novo, if you like. So a virus can evolve to be able to infect me using my machinery, but it's just a small piece of DNA or RNA that has popped out and has found a sneaky way of cheating by not having to go through my germ cells to get to the next generation. Yeah. But so I'm really looking, I mean, in my lifetime, I hope that the missions to Enceladus, the frozen moon, Saturn, are successful, and they managed to drill. No, they're going to fly through there are ice. Enceladus is covered with ice. Underneath the ice is miles, kilometers of water, very salty water. And there are ice volcanoes on Enceladus which spew this frozen water, this water into space. It freezes. And so if there's life on Enceladus, it's the best place for it to be. It's warm under there. The water is liquid. It's salty. A spaceship is going to fly through the plume of one of these volcanoes and come back to Earth. And so before I'm done, I would really like to know if life on Enceladus uses the same genetic code and genetic material as life on planet Earth. I don't know which way to bet at the moment. The excited bit of me wants to bet that it's completely different. And then the boring bit of me wants to think that, yeah, maybe it's just a bit knocked off the Earth once and floated all the way to Enceladus. Or maybe it's life started on Enceladus and floated here. Anyway, so those of you who are younger than me, watch out for the reports from what comes back from Enceladus. That'll be fun.
You paint an enticing picture of like warm, salty water. So how could there not?
Unfortunately, you're being irradiated by anyway. It's not all warm and salty.
Just slightly irradiated. Yeah. Do we have any other questions in the room? Yeah.
Yes. Yeah. So, resurrection biology, do I approve? And is it a good thing? So from a purely digital sequence information point of view, we want the Library of Life to be open for use, and we are not a priori going to have any control over what that's used for. So somebody could use it to invent a new virus which kills people who disagree with me. You know, and that's a horrible thought, but bad actors happen. So there is an issue about having an open library of life, that one can imagine ways that that could be used wrongly. But I think just like the open any library will have books in it which might have passages in them which can be taken out of context and used to hurt people, I think in the same way we would do that by our society policing what goes on. So I think that's how we do that. In a specific question about resurrection biology, I'm not interested in resurrection biology as it is currently practiced by Colossal or whatever. I think it would be much more interesting to try and conserve some of the astonishing species we have, and much more interesting to try and use that immense knowledge that the people working in Colossal have to something that's slightly more socially useful. I mean, I don't think resurrecting the mastodon is going to protect the taiga forest from something. I don't know. the minute a mastodon is created anyway, it's going to be the most endangered species on the planet because there's only one of it. So it's going to be like white rhinos again. So I'm not particularly entranced, even scientifically curious about resurrection biology. We've done a bit of our own resurrection biology, actually, with viruses coming to this. So when a bit of permafrost melts up in the taiga, out of it comes things which are still alive, because there are lots of things alive today which you can freeze. Things that live in Arctic regions, things that live up mountains, are used to being frozen. And so they have what's called extremophilic, so cryophilic, cold-loving biology. So there's lots of things you can freeze and bring back to life, and freeze and bring back to life. And so when this taiga permafrost defrosts, if you do it in a very clean way, so you cut out a piece of the ice and stick it in a sterile environment and let it melt, you can find living organisms in there. So nematodes, hooray, and rotifers, and there are also bacteria, obviously, and also giant viruses, which are terrifying because we don't know what they do. So that's the sort of resurrection biology I'm more interested in, is discovering something about life that lived 40,000 years ago by defrosting it from the ice. We sequenced one of those nematodes, of course. So.
I think we're rapidly running out of time, but there was one question that came in online that might be a good one to actually finish on, which is, so this is all very, very kind of technical, highly detailed scientific information. How do you put this into practical use for conservation? Like, how can you take this out into the field and do something with it?
So I'll do that by giving an example of conservation use of the genome. So otters in Europe, until they were formally protected, were really persecuted, and populations were dropping all over Europe, and especially in Britain, where the number of otters in Britain dropped into the low thousands. This is in the 1950s, and they were protected in 1960. And since then, they've rebounded. And so there's a real question about what do we do about these populations? So should we mix them? So there are little relic populations in Cornwall, in Wales, in Scotland, and in East Anglia. Do we allow them to mix? Or there's something special about their adaptation to Cornwall, Wales, Scotland, East Anglia that means we should preserve them separately. So how should we promote otters? I think everybody agree promoting otters is a good idea? Okay, so how do we promote otters? And so what we did, we sequenced the otter from a roadkill specimen, of course. This is traditionally, apparently, with the Otter Project in Cardiff, who have freezers full of roadkill otters. Anyway, and so what they've done is they've gone back and done genomics on lots and lots of their otters. And they've discovered that the populations in Wales and Cornwall are actually quite closely related, and have approximately the same pattern of the population going down and down and down until the 1950s and rebounding slowly. Population in Scotland is very different. The population in Scotland never was actually very small. It was probably undercounted, and it's been was much less under threat than the Welsh and Cornish otters. So that's a different, but it's quite genetically distinct. The third one was looking at East Anglia. So the population in East Anglia has exploded in recent years. There's lots of otters in East Anglia, but they discovered the otters in East Anglia are really strange. And what's probably happened, so they have a whole bit of genetics going on in East Anglia, which is different from the rest of Britain. And they seem to be linked to the Balkans. So Eastern Europe. So probably what's happened in East Anglia is that there were petting zoos or little local zoos, which had well-fed otter mums and dads in them, who had litters of 10, 15 kits, which is not too difficult. And this was great. And then lots of people came in and they fed them lots of fish. And then they got to winter and they thought, actually, we don't have enough space in our zoo to look after 30 otters next year. And then you do the maths and you go, okay, and 130 the year after. And so it looks like probably these are releases from captive populations into East Anglia. And so that's why the genetics is weird in East Anglia. So there is now a real clear demarcation towards how you deal with otters in East Anglia, which just let them go, and how you deal with otters in. So that's a direct impact of us having the genome before people had looked at little DNA markers like DNA barcodes, and they'd sort of found four groups, but they couldn't actually say anything about where those groups came from. So, yeah, so
Excellent.
that's one example. Keep going.
Great. Well, I think
Eagles, squirrels, white-tailed sea eagles. They've been used to develop new quotas for fish stocks in the North Sea. So anyway.
I think on that endless list, I think that is a good point to end. So can we all give Mark thanks again, please?
Thank you. I just want to say, those of you who took a photographic QR code at the end, this is the correct QR code. The other one was the wrong QR code. Oops.